Hello, dear students. So good morning. I guess we can start. So only 57 students joined, but I guess in 5 minutes I'll add more students after 5 minutes. So this all students will be missed. So and got absentees. Okay. What should we talk about? In last lecture, we discussed the P53 gene, so regulation P53 gene and how P53 gene takes part in cancerogenesis. And this cancerogenesis process, you see, it depends on a lot of mechanisms, and so I'll remind you. So I'll remind you this diagram. So this we have there. It depends on growth factors and growth inhibitors. Yeah, we have growth factors and growth inhibitors, and growth inhibitors they affect process before the P53 gene and after the P53 gene activity. It's after P53 protein group activity, and you see and growth and damage of these inhibitors might cause the activation of S phase of proliferation and loss of inhibitory functions in cancerogenesis. They can cause the overproduction of cells, proliferation of cells, unstoppable proliferation of cells. Okay, but it's not only depends on growth factors. Yeah, so now we discuss the growth factors and how they affect it. We also discuss what the retinoblastoma gene, RB gene, and damage of retinoblastoma gene. So also affects the growth of cells, and it causes cancerogenesis. But you remember we have a lot of mechanisms to prevent cell growth from the okay. We have today we'll continue with the transforming growth factors. Transforming growth factor beta pathway. So in easy words, so transforming growth factor is produced by autoimmune response. So due to secondary so alternative pathway of macrophage activation, TGF beta is produced, and due to this TGF beta, so angiogenesis and other and stroma development happens, which serves to improve the activity of malignant cells. Growth helps to grow for malignant cells. So let's talk about okay. That's we discussed it a little bit. Yeah, and one more very important function is TGF beta signaling. So TGF beta signaling is it causes a epithelium mesenchymal transition. Yeah, once again, cancer is a carcinoma. Yes, so epithelial cells, and they have attachment to one cell to another. But this epithelium mesenchymal transition, which is activated by this, it causes later promotes migration, invasion, and metastasis of tumors. That's the role of TGF beta. So that's in one of features of malignant tumor is it's a feature to facilitate invasion, metastasis, and other things which discuss later. Okay, and we have contact inhibition. You remember that especially for epithelial cells, so you know that every cell is attached to another cell. Every epithelial cell is attached with the tight junctions, and it happens with help of E-cadherin. And so one of the best examples is E-cadherin. Okay, and epithelial cells, they have VNT receptors on the surface. And these VNT receptors, they can be stimulated by some growth factors, so VNT, and it can cause a proliferation even if joining of neighboring cells is happen. So when joining is there. And just imagine, so just main idea of this point. Yeah, the epithelial cells. So while epithelial cells are adjacent to each other, they have connection between each other. And so it means that surface of epithelial cells is integrity. So we have integrity of epithelial cells, and we don't need to reproduce it. That's why, so when we have a normal connection, this we have different proteins, and one of them is well known as APC. So when VNT receptor is silent and APC, so it stops the activity of beta catenin, which is attached to E cadherin. And even if somehow beta catenin getting free is getting free from ecadherin. So this APC, so it envelops it and destructs this beta-catenin if it's not in large amounts present in the cytoplasm, and this beta-catenin can provide no function. But when injury to cell happens, now, when injury to cell happens, these E-cadherins are detached, and then detachment of E-cadherins causes the beta-catenin liberation, and along with the VNT signal, so due to injury, cell injury, this beta-catenin become the activator factor. So this it causes the activation of genes which cause the mitosis. So that's one another mechanism of mitosis, and this beta catenin, if it's not silenced, so we have proliferation, and VNT stimulation can cause the proliferation, and just damaging of cell-to-cell attachment can cause it. Okay, Sadula. So here you see it signals and VNT signals, they interrupt the activity of destruction complexes which made with the help of APC. So what is APC? So don't never mind. So it's some protein in the cytoplasm. And this beta catenin is like activator factor for transcription of genes responsible for S phase activation and other promoters of mitosis, promoters of cell cycle. You see, it's beta catenin, so it's special for epithelial cells. Yeah. Okay, but what happens in cases of mutated APC? So APC is mutated, and epithelial cells are integral. Yeah, so beta catenin is released in very small amounts here, but this beta catenin cannot be interrupted like shown on this resting cell, and this beta catenin acts like so in tumor in this with mutated APC. This beta catenin anyway activates this gene expression here, and it promotes the mitotic signal. So you see, APC is not absent here. APC is just mutated and cannot provide proper function, and that's why it causes the proliferation. So normally it happens only when cells are when we have injury to epithelial cells, but here it causes but here we have no epithelial cell damage, but anyway it causes the proliferation. It's a very common pathway of tumor genesis of tumor. Okay, and here we stop the genetics. Yeah, here we stop the genetics of this of tumorigenesis, of cancerogenesis. That's why let's switch to the altered cellular metabolism. So altered cellular metabolism. So here you see that in 1931, Otto Warburg. So he got Nobel Prize because he described the Warburg effect. The Warburg effect we discussed a little bit yesterday with Hamza, if I'm not mistaken. So we discussed it. He asked me about this, and I explained it in a little way. So you see, let's talk about this how Warburg effect happens. Warburg effect is not only happen in cancer cells. Warburg effect is common for every dividing cell. You see, every dividing cell requires a lot of building blocks, building bricks, so which are necessary to produce new cell, which are necessary to produce new matrix, necessary to produce new organelles. So every dividing cells they require some new building blocks. Okay, but question. So let's discuss very generally. So what is a question cell? So here we have okay, growth factors. So site of activation of growth factors. So if it's silent, so cells shouldn't divide. And so glucose intake to the cell goes very slowly. Even sometimes this glucose transporter is closed. Yeah, because it doesn't require energy here. And cells without activation of growth factors, so they start to undergo process known as autophagy. And autophagy will destroy some organelles inside the cell, uh, and this components of inside the cells, they are used for synthesis of energy. And which energy is used here? Of course, so lipids and amino acids catabolism. From entire inside the cell, they enter into the Krebs cycle. And Krebs cycle is used totally, totally used, and it causes production of ATP and CO2 only. That's what you used to discuss in biology or biochemistry. That that shouldn't give you questions. Yes, this is normal mechanism of growth, or mechanism of cell survive. So ATP and CO2 production, and we don't secrete some building blocks because cell shouldn't divide. And actually, for example, in case of muscles, yeah, when the muscles are not growing, and in brain, yeah, we don't need to produce new building blocks. We predominantly we produce just ATP because these cells are very active. But in growing cells, they use a less amount of ATP. They don't need too many molecules of ATP here, and instead of it, we use so it starts to use everything which it can get. So cancer cells, normal or tumor cell, that doesn't matter. So growing cell, they intake a lot of nutrients like glucose, even lipids, which are not shown here, but okay, glucose is intake inside the cell, and this glucose is used to produce directly from the from without the use of mitochondria. It directly can cause the production of some nucleotides, amino acids, and for synthesis. Yeah. Uh, so but lactate is produced. Yeah, that's why inside the cell we have a little acidic environment. And sometimes this acidic environment inside the cell might cause the death of the cells or growing cells. But usually it's regulated very well, and this lactate is coming into the mitochondrial Krebs cycle. But unlike a normal Krebs cycle, instead of ATP and CO2 production, here it causes the production of some components. So it's escaping from a Krebs cycle, causes the formation of a lot of amino acids. So all the replaceable amino acids you discuss in biochemistry, they are derived from mitochondrial metabolites. So during the mitochondrial activity, and also a lot of lipids are produced. We need these lipids for production of cell membranes, for cell division, for endoplasmic reticulum, or so other organelles. So they're required, and that's why in mitochondrial activity we produce these components, and and you see very little amount of ATP is produced. They're produced predominantly by glycolysis, anaerobic glycolysis, before the entering into the mitochondria. And in mitochondria, so it doesn't need oxygen, and it produces nucleotide, amino acid, and then from amino acids produce a protein synthesis, and it happens under the growth factors, and activation of these growth factors causes Warburg effect. Okay. Um, then also, yeah, don't forget we have glutamine, also amino acid glutamine, which also used in our metabolism. And they are promoted by growth factor activation of the cell, and by mechanisms we discussed before. Our tumor cells are able to promote, to produce their own growth factors, or they can use growth factors. They can use other growth factors which are present here. And anyway, this effect, Warburg effect, is activated here. and it explains a lot of features of tumor. Tumors are not very so usually tumors, malignant or benign tumors, they are very sustained. They can be sustained without even without oxygen intake. They don't need oxygen. They don't spend oxygen of our body. They spend nutrients of the body, but not oxygen. And that's one of that is one of feature why during the tumor we have a process which is named as cachexia. Yeah, cachexia, because all the glucose and glutamine which present in the blood, so it takes inside the cell and used for proliferation of the cell, proliferation of these types of cell. So that's why and it gives a additional feature to the cells, to growing cells, because here we have less reactive oxygen species, because mitochondria doesn't use it, and mitochondria so just utilize it in easier way, and so don't intake. That's why these cells are they can grow, and then without cell death. This is oncologism. Autophagy, we discussed what is autophagy. So it's used in normal cells. So in case of nutrient deficiency, so and in cells not only arrest their growth but also cannibalize their own organelles. Yeah, but it never happened in it never happens in tumor cells, because we don't need autophagy here. Autophagy is dumped in tumor cells. Okay, so here mechanism happens. So it's due to mutation happens, or might be so a lot of pathways. So it's even without mutations, so Warburg effect happens. But sometimes even mutation in isocitrate dehydrogenase happens, and which doesn't allow to produce a lot of ATP. Here, instead of this, produced hydroxyglutarate. Hydroxyglutarate, it's a typical oncometabolite. It stops the epigenome regulatory enzymes, and it causes altered cancer gene expression, and it causes cellular transformation. So that's a metabolism of cancer cells. So it's very broadly explained. So we need to continue. And then we have evasion of cell death. I said that tumor growth need to evade the cell death. So, and which type of cell death is we are talking about? It's of course apoptosis. We need to prevent apoptosis by any way. We need to prevent apoptosis, and one of these effects we already discussed. For example, p53 gene. So if we have p53 gene pathology, I mean alteration of p53 response. So of course DNA damage will not cause the activation of Bax and Bach family proteins in mitochondria, and it is not followed with cytochrome activation. It's one feature. Okay, it's BCL2. So especially BCL2, we are talking about BCL2. Okay, and normally deficiency of growth factors and survival signals outside the cell, they also can cause apoptosis and activation of BIM and other sensors, which also cause the activation of Bax and Bach family. And this Bax and Bach family, so they cause the releasing of cytochrome C, and other so like APAF, yeah, so apoptotic bodies are produced, and they cause activation of cascade, and finally it leads to apoptosis. Yes, so this is normal effect, but injury to p53 response so cause the blockage of apoptotic signaling. Okay, but even if growth signals, deficiency of growth signals happen, so we have a feature. So you see, in some tumor cells, instead of damaging of this gene, of this protein pathway, they start to increase amount of Bax and Bach family. So, and in a second set of lectures in cell death, we discussed that Bax and Bach family proteins, they are inhibitors of apoptosis. Just block the cytochrome leakage from the mitochondria, and some tumors, they are able to overproduce this Bax and Bach, and these cytochromes are not released. and they're like blocked. And even if we have p53 response due to mutation or any other type of DNA damage, so it causes so it doesn't cause a leakage of cytochrome because of BCL2, BCL-XL, and MCL1 overexpression happens. Okay, and also we have inhibitors of apoptosis, so which are specifically binds with the caspase 9 and which interrupts the apoptotic process. So this is also there are also mechanisms of blockage of apoptosis. That's and possibility of this blockage allows it to grow constantly. Because I said before, yeah, that during the oncogenic DNA changes, so due to active cell growth, active cell division, so a DNA damage is inevitable. So anytime any cell division is followed with DNA damage, and in rapid cell division, this DNA damage is going very tremendously. So and a lot of changes happen, and that's why we need to block apoptotic signals in our body. That's evasion of apoptosis happens. So here you can read about it. So in an easier way, it's explained in this chapter. Then, limitless replicative potential. You remember that in our cells, we have limitation of cell division. So usually it's they say that it's 70 doublings we have. So only no more than 70 doublings. It's a lot. It's a very large amount of divisions, and usually it's enough to survive until 100 of years. Yeah, but for tumor cells, it's not enough, because so this division happens. So one division a day might be one division in 8 hours, and of course, so very soon, if this rule will be handled, then the 70 doublings will be interrupted. So it will interrupt the normal growth of tumor cell. So normal, I mean, in brackets, yeah. But they need to reproduce themselves to make it tumorous, and it undergoes these changes. You see, multiple cell divisions, they cause a shortening of telomeres. I remind you that telomeres are present on the spikes of short and long arm of chromosome, and when it's very short, so it's like an anchor it's like an anchor of gene, so which we don't afraid to lose, during the cell division. If you don't afraid to lose it, if you don't afraid to lose it, like here, for example, like in stem cells, we have it. So this it undergoes a cell division. But if it's very shortened, so then p53 mechanism activated, p53 proteins activated, and then leads to senescence of the cells, and these cells are senescent and remain stable. But if p53 gene is interrupted, like we discussed before, this after the cell division, so these telomeres, they get very short and too short, and that's why sometimes without the telomeres, chromosomes get attached to each other, and it forms the super chromosome. Yeah, so super chromosome, fusion of chromosome, and sometimes and it undergoes, and when it undergoes the mitosis, it divides into these fractured chromosomes. And you see, in chromosome sequence, and in tumor cells, usually in cancer cells, we can't find this picture that 46 chromosomes are present in our cells, in our body. No, it's not 46. It might be more or less, and even this and these 46 are not similar to 46 chromosomes of normal cell. And you see some fracture of one chromosome will be attached to another chromosome, some fractures here, and then you see, and it undergoes this pathologic cell division and without interruption. Some genes are lost during this type of division, and due to this, a lot of cells died. and that's why this damaged chromosomes sometimes it causes the mitotic catastrophe. It's mitotic catastrophe, and cells died. And a lot of tumors, potential tumors which present in our body, they are died. So even with all these changes we discussed before, all this p53 gene damaging or others, other processes which are damaged, it causes mitotic catastrophe, and then we don't mention it. Yeah, so we as humans, we don't mention it. We have no tumor and we are happy and healthy. But sometimes during these chromosomal changes, it causes genetic changes, and it might activate the telomerase. And this is catastrophe for us. It is good for these tumor cells, but it's catastrophe for us because telomerase it is a enzyme which build up a new telomere sequences about and this telomerase will give a constant ability of this tumor cells for division without interruption. So you see these cells became like a stem cells, like here this is a stem cell, and the same stem cells, not same, but they're a little different because of this changed chromosomes are present here, but still they are preserved, and new chromosomes are produced here, and they're preserved by telomeres. Telomeres are very built up here again, and now cell division happens normally, and without interruption it might happen, and even most of genes are preserved in these cancer cells, and that's very dangerous, yeah, for us. So that's lengthening of telomerase, lengthening of telomeres. Yeah, we discussed it. Okay, then sustained angiogenesis. I already said, yeah, that angiogenesis is sustained, so especially it happens with the help of PDGF and or insulin-like growth factors, and this newly formed blood vessels are very small, very gentle, and that's why I already said it to you in lecture that the little damage to cancer cell to cancer, so for tumor, so will leads to bleeding. It leads to bleeding, and this bleeding is very hard to interrupt because blood vessels are very small, they have no smooth muscles around, and that's why the smooth muscles don't have activation of, as you remember, endothelin. They produce endothelin, but smooth muscles are not surrounding it. That's why they don't cause a collapsion of these blood vessels, and that's why it causes profound bleeding sometimes. So that's that's the danger of cancer, that it causes bleeding, hemorrhage. Okay, but you see, and that's actually the point of no return sometimes for benign malignant tumor. So benign tumors, they usually don't have angiogenic switch. So we have this angiogenic switch. So benign tumors, they are unable to produce PDGF, platelet-derived growth factors. They are unable to produce fibroblast growth factors or VEGF, vascular endothelial growth factors. Might be produced, but in very little amounts. Yeah, but if some genetic changes happens in this benign tumor, and they are able to produce this PDGF and VEGF, it is known as angiogenic switch. And this angiogenic switch so will determine the formation of malignant tumor. And now from this after angiogenic switch, it is malignant tumor. Anyway, it's malignant tumor, and they can use their own VEGF. They can use the macrophages which are present here, and alternative activation of macrophages or activation of fibroblast, which produces growth factors. They can cause this vascularization of the tumor. And I remind you that vascularization of tumor causes the increased amount of nutrients which come to the tumor cells. And now the cells will, especially along with the Warburg effect, they're able for cell division. They're able to produce a lot of building blocks, a lot of cell membranes and nucleotides, and then continue division this way. And another idea, so this so yeah, some example. For example, p53 stimulates expression of angiogenic molecules such as thrombospondin 1 and repress expression of pro-angiogenic molecules such as VEGF. This loss of p53 in tumor cells provides a more permissive environment for angiogenesis. You see, it's also related. So it's not only coincidence. It's some kind of also regulated by p53. But if we have damage to p53, we have lesion of gene TP53. It interrupt the vascular endothelial growth factor expression, and this VEGF typically increases the tumor in patients. Okay, a little bit relax. So I want to say one example from movie I had. So I seen a movie, and in one movie, so when might be heard Doctor House. Yeah, that's very interesting series where doctors given the very strange diagnosis. Yeah, so they give a diagnosis to patients with a strange appearance, with symptoms which are don't match to normal diagnosis. And in one series, there was a patient which has all symptoms of tumor, but they can't find the tumor. So all symptoms of tumor happened here. So we didn't discuss it, but we have paraneoplastic syndrome. It's a syndrome when our own immune system fights against our own cells because we had tumor. Yeah, but this tumor, not only it fights not only against tumor but also against our own cells, because tumor is derived from our own cells, and that's why antigens of this tumor might be similar to our own antigens. And that's why immunity cells they start to kill not only tumor cells but also our normal cells. So what's the point? And tumor was very small, but it produced a lot of adverse effects, a lot of bad effects. So that's why, and but they don't know where this tumor is located, and what they start to do. And they decided to give to patient VEGF, vascular endothelial growth factor, intravenously. And it causes the proliferation of blood vessels, and especially blood vessels will be proliferated in place of tumor. And due to proliferation of blood vessels in tumor, so it causes proliferation of tumor. Then they could understand so where this tumor is located. So it's don't use it in your practice, so because it's very irresponsible, because you will promote not only angiogenesis and growth of tumor but also its dissemination, because when we have more blood vessels in tumor, so it increases the possibility of metastasizing. Yeah, and metastasia might spread it. But just in mass culture, I explained you one example of mass culture when we can say that VEGF promotes the tumor growth. Okay, then invasion and metastasis. So then now finally we have to talk about invasion and metastasis. A little bit we already discussed. Okay, let's talk about definition. Invasion and metastasis is a major cause of cancer-related morbidity and mortality, result from complex interactions involving cancer cells, stromal cells, and the extracellular matrix. So invasion. So what is invasion? So invasion, it happened because of loosening of intercellular connection between tumor cells. So we discussed before, yes, that EC adherins, which bind to B-catenin. So when we have loss of this EC adherins attachment, this process is known as epithelial-mesenchymal transmission, and it causes mutational inactivation of E-cadherin genes, activation of beta-catenin genes, or inappropriate expression of snail and twist transcription factors, which suppress E-cadherin expression. So anyway, E-cadherin, which is playing role in attachment of cells here, for example, as you see here, in like tight junction. So loss of this cadherins, E-cadherin, it causes the detachment of one cell to another. Yeah. Then you see, and this, okay, loss of intracellular junctions happen, and then cells start to produce collagenase, type 4 collagenase. So how it happens, we don't know, we don't totally know, but somehow type 4 collagenase is produced, and basement membrane is composed of collagen type 4. Yeah, and basement membrane usually don't allow the passing of epithelial cells under it, but activation of type 4 collagenase causes the invasion of these epithelial cells in adjacent subepithelial loose connective tissue. This is invasion, and that's how it pass under the layer. Okay, also we have this autocrine motility factors, which cause so in some tumors we have a autocrine motility factor expression, so due to which cells can easily migrate in a subepithelial layer, subepithelial in under the basement membrane, and then cells is this process of invasion happens. So, and the second step, when invasion happens, these transformed cells, when they invade the basement membrane, it comes into the connective tissue. It's invasion. But when the same invasion continues and reach the blood vessels or lymphatic capillaries, doesn't matter. So it goes. They can evade even this capillary. Doesn't matter. Once again, doesn't matter. It's a lymphatic capillary or a blood capillary. It invades it and enters inside the blood vessel. And then this tumor cells, they of course they are they activate the von Willebrand factor, and due to this platelet aggregation happens here, and it forms something like tumor cell embolus. Tumor cell embolus, and this tumor cell embolus can travel along the blood or lymphatic capillary in other places. So if it is a lymphatic capillary, it travels into the lymph nodes. If it's a blood vessel, so it travels all over the body. Yeah, and it spreads, and spreading of tumor cells, tumor cell embolus goes by the same features as any other embolus spreads out. So here I want to give you examples from embolus. For example, if we have a tumor in, okay, for example, tumor appeared in the uterus, in female's uterus, yeah, and then it enters into the blood vessels of uterus. So where is the first metastasis we expect to find? So where is the first? So maybe you can answer, guys. You can answer now. From the uterus, where blood?
Yes, sir.
Where blood comes through the uterus? After the uterus, blood. So where veins of uterus come later? So where is this embolus can stuck? The next.
Sir, mm,
Urinary bladder.
Where?
After ureter, sir, urinary bladder.
No. Uterus, not urethra. Uterus. So it enters into the blood vessels. So take a look in the blood vessels. So where? So you remember the anatomy, yeah? So you remember anatomy, and where blood goes from the uterus, and where blood goes? It goes directly into vena cava inferior, but vena cava inferior is a large blood vessel. Then out of this large blood vessel, it enters into the heart. Then in the heart, it's also a large blood vessel, and in the right heart, yeah, in the right atrium, then in the right ventricle, and then from right ventricle it enters into the pulmonary arteries, and from pulmonary arteries it spreads splits into small capillaries of lungs. This capillary and this embolus can stuck in lungs. Yeah, and that's why we first metastasis. So first far metastasis we have in after the tumors in uterus. It's a lungs. It is a lung. So by the same way, from prostate tumors might appear there. From the same feature, tumors of so for the skin tumors, they usually have metastasis in a lung. Okay, for example, but if we have a colon cancer, yeah, and spreading metastasis of colon cancer, where would they go? So where will be the next metastasis after the colon tumor? Any suggestion?
Sir, blood in the stool, we will notice.
Hmm? Mahnur again?
Sir, again, can you colon? You are talking about colon cancer.
Colon cancer, yeah.
Sir, we will notice blood in the stool.
So it's not metastasis. Yeah, blood in the stool will be there. Yeah, it's a symptom, but it's not metastasis. Yeah, it's not about metastasis. I'm asking about blood. Vascular metastasis which happened. So again, you didn't understand my question. You see, it's a colon cancer.
Sir, liver metastasis will happen.
Absolutely right, Mahnur. So now you understand my question. So yeah, liver metastasis will happen because of what? Because colon is a part of a portal vein system. Yeah, portal vein system, and from portal vein it enters into the liver and splits into the sinusoids. Yeah, and the sinusoids, and from the sinusoids, this tumor cell embolus will stuck there. Will stuck in the liver, and we have massive metastasis in liver, and that's the most common type of metastasis. But if we have a tumor in a lungs, yeah, so where would we expect to find metastasis? And answer is everywhere, everywhere. So all over the body would be found metastasis if we have cancer of lung cancer. So we can it might be so first of all it is found in brain, metastasis in brain. So also if we don't talk about local invasion, if we talk about far metastasis, would be found in the brain. Okay, but you need to think here so in this way, so that the spreading of tumor goes not only by blood vessels, not only by but also it goes by lymphatic capillaries in lymph nodes, and then from lymph nodes and also it has a local invasion. So, for example, in lung cancer, in cases of so one example we will discuss it in the respiratory system, we have Pancosta tumor. It's known as Pancosta tumor, which appeared in the upper lobe of the lung, and tumor of this spreads so very rarely spreads in blood, but it spreads by invasion, and it invades into the axillary nervous nerve bundle, and it causes a pain in the hand. So it causes a chronic pain in hand, and that's why sometimes, not always, but sometimes pain in the hand is a symptom of cancer. That's why it might be you heard about this. So it was very good joke. Yeah. So never Google your symptoms. Yeah, because sometimes even pain in the hand, so it might be a symptom of cancer. But you have to understand that sometimes it really happens. Rarely, but it happens. Okay. So, and you see, now to make this tumor cells motile, so we need to undergo these changes. It's a local degradation of the basement membrane. Then we have changes in attachment of tumor cells to extracellular matrix proteins. Yeah. And also locomotion is a final step of invasion. So locomotion, when the cells starts to produce here autocrine motility factors, and they can transport along with the fibronectin, and this is invasion. Okay, vascular dissemination and homing of tumor cells. So, as I said, metastasis, they can spread all over the body with the help of blood vessels. The site at which metastasis appear is related to two factors: the anatomic location and vascular drainage of the primary tumor and the tropism of particular tumors for specific tissues. So, for example, some tumors they are unable to adhere in some organs. For example, a lot of brain tumors they can't find the nourishment there. They can find the nourishment in other locations and in lungs, and they don't have a metastasis. But sometimes it has. So yeah, for example, here you see bronchogenic carcinomas tend to involve the adrenal glands and the brain. Prostatic carcinoma preferentially spreads to bone, so that's typical location. Neuroblastoma spread to liver and bones. So that's it's tropism. We have tropism, so that's why it's not only we don't need to think so. Yeah, every time tumor when tumor spreads by blood, it can comes to this area. But don't forget that we have tropism. Such organ tropism may be related to the following mechanism. Tumor cells may have adhesion molecules whose ligands are expressed preferentially on the endothelial cells of target organ. So that's one of idea. Chemokines may have an important role in determining the target tissues for metastasis. Okay, so some chemokines are produced. Okay, that's not important. In some cases, the target tissue may be a non-permissive environment, unfavorable soil, so to speak, for the growth of tumor seedings. For example, also there are well-vascularized skeletal muscles and spleen are rarely sites of metastasis. You see, skeletal muscle and spleen are very rarely sites of metastasis. So let's talk about okay, metastasis. We discussed what is metastasis. It's continuation of growth in some specific tissues where they attach to. Okay, and then and last so one of the last things we discuss about the tumor, it's evasion of immune surveillance. So as I said before, a lot of tumors which match to all this criteria we had before, they can be killed by our immune system, like NK cells, because of changed gene expression, because of changed MHC, because of changed HLA. So our immune cells can be killed. For example, we have specific tumor antigens. Cancerous disorder that is caused by driven mutations in oncogenes and tumor suppressor genes, which in most instances are acquired rather than inherited. And we have all of these mutations may generate new protein sequences, new antigens that the immune system has not seen and therefore is not tolerant of and can react to. So that's the main idea. So because of genetic changes, acquired genetic changes produce different antigens, oncoantigens. Some of this antigen is a tyrosinase, an enzyme involved in melanin biosynthesis that is expressed only in normal melanocyte melanomas. It may be surprising that the immune system is able to respond to this normal self-antigen. Um, and if it's produced in small amounts, so it's not recognized by immune system. But if it's overexpressed, so our immune system um, fails to induce tolerance, and then our body fights against this tyrosinase. Other we have cancer testis antigens. Cancer testis antigens. So I remind you that in testis we have a barrier, blood testis barrier, and this blood testis barrier is maintained by tight and gap junctions, and it prevents the immunity cells entering into the inner environment of seminiferous tubules. Yeah, and that's why because of what? Because there we have germ cells, and these germ cells they can express different genes because there we have meiotic process, yeah, and due to recombination, due to meiotic process, they can express another genes. And that's why MHC1 molecules there are a little different from other cell other cells of the body. And that's why if they will be revealed, so in case of tumor or in case of injury of testis, yeah, so might be just a fight, yeah, in just a fight. So when it's sorry, when it gets injured, and our immunity can attempt to kill our germ cells, and it causes infertility sometimes. So sometimes, if tumor is produced with help of Epstein-Barr virus, and we have a lot of studies which says that Epstein-Barr virus and human papilloma virus, they can cause some tumors, and then cytotoxic T lymphocytes may detect these antigens, and they kill the tumor. It kills these infected cells, and some of them potentially are tumor cells. But anyway, they kill it like viral infected cells, and didn't realize that it possibly could give a cancer for us. Um, okay, and that's why Epstein-Barr virus. So it causes the uh, Epstein-Barr virus-related B-cell lymphomas occur at significantly higher rates in individuals with defective T-cell immunity, such as patients infected with HIV. So you remember that in HIV, so tumors are also very promoted because of antiviral activity is dumped. Yeah, and this Epstein-Barr virus, they can cause B-cell lymphomas. And you remember the diagram as shown in the first lecture about about widespread of diseases in some areas. You can find that in Africa there are a lot of countries which predominant type of cancer, predominant type of malignant growth is a B cell lymphoma, because it happens only with along with HIV. Okay, effective immune responses to tumor antigens. Okay, here let's take a look. Here, this is normal. That's normal what happens. So you see, this is tumor, yeah, and then our cytotoxic T lymphocytes, CD8 positive T lymphocytes, they detect it and kill it. So, but to detect it, we have to be sensitive for this, and that happens this way. Then dendritic cells swallow some residues of these dead cells of tumor, and they transport it into the lymph node. In lymph node, these dendritic cells they activate with their MHC1 major histocompatibility complex type 1 class 1. It presents this antigen to T lymphocytes, CD8 positive T lymphocytes, and the CD8 positive T lymphocytes they traveled to the site of cancer, and they start to kill it by direct CTL, so it's by direct cytotoxicity, and that's a normal immune response against tumor. But you can say, so why not all the tumors are digested? So normally, sometimes um, dendritic cells they require. Mhm. Yeah, so they require co-stimulation of CD28, and B7 might activate only or CD28 or CTLA4. If we don't require co-stimulation, so it activates directly here. But if it requires co-activation of this, we have anti-CTLA4. This is a drug which is used, and CD8 positive cells promoted to form cytotoxic T lymphocytes. That's normal activation. But what happens here? Inhibited CD8 positive C cell T lymphocyte. It has a how to say it right. So normally, sometimes PD1 is not matched to PD1 ligand of the tumor cells. Yeah, and this coactivation it causes tolerance. Yeah, it causes a tolerance. So when it's attached, so it causes a tolerance. But and they inhibit the cytotoxic activity of this T lymphocyte, and that's why new drug is produced. So new drug which is made by, oh, I forgot the names of these Nobel Prizers, but a Nobel Prize is given to this scientist. They found the way how to block this PD-1 and PD-1 ligands. So we found antibodies against it, and then we have no interruption of signal. And these tumor cells, they don't give a how it's called, tolerance to it. These tumor cells became intolerant, so in our cytotoxic T lymphocytes became intolerant to these tumor cells, and with the help of added antibodies. And due to this, the killing of cancer happens, and that's one of the most modern way of cancer treatment. So, and actually, it has a lot of we have a lot of hopes to this treatment. So we can, if we can block this PD-1, a PD-1 ligand on tumor cells, it can cause the easy killing of our cells, even if it is a fourth stage of cancer when it already has metastasis, because our immunity already recognize these cells, and we can digest it by our own immunity system. So this is anti-PD-1 and anti-PD-1 ligand antibodies. They can be injected intravenously, and then and everything else is made by cytotoxic T lymphocytes. Okay, that's the immunity point. Then here we have some examples. So genomic examples, so which changes we have. So then etiology of cancer, carcinogenic agents. So what causes changes? Okay, you remember this avoiding of immune destruction. So let's talk about this. So in general, now we just collected together in one diagram. So okay, avoiding growth suppressors. Avoiding growth suppressors. So you see, we need to inhibit cyclin independent kinase inhibitors. Yes, cyclin independent kinase inhibitors, they should be inhibited, and then growth of tumor continues. Yeah, but if we activize the cyclin independent kinase inhibitors, so this growth suppression happens. It's a therapeutic targeting. Yeah, so we can work on each of these steps. So then in enabling replicative immortality, yeah, so we have in a replicative immortality of the cells because of telomerase, yeah, and but if we give the treatment like telomerase inhibitors, the cells will be unable to produce to elongate their telomere, and then these telomeres are not elongated telomeres, they will cause a mitotic catastrophe, and cells will die. Okay, then another feature, so tumor promoting inflammation. So how we need to increase the inflammation, so we need to decrease inflammation in the sites of tumor, and we have selective anti-inflammatory drugs to prevent it, because inflammation for tumor is good because it causes vasodilation, it causes the formation of new blood vessels, it causes chronic inflammation which gives attempts at healing. Yeah, and that's why this inflammation is not very bad for our cancer cells, especially if they can avoid the immune system. So that's why we need to administer to this patient anti-inflammatory drugs. Okay, and then activating invasion and metastasis. So these tumor cells, they active their metastasis, and we can inhibit this growth factor. Yeah, so hepatocyte growth factor, CMET. So doesn't matter. We need to prevent epithelial mesenchymal transition. So genome instability and mutation. Genome instability mutation might be dumped by MDM2 inhibitors. So we just okay, we skipped this information. Okay, but keep in mind that we can affect the genome instability too. Inhibitors of VEGF signaling. Yeah, so angiogenesis is induced by the tumor cells, and we need to depress the formation of blood vessels. So and that's why we can use inhibitors of VEGF. Also, it's very usable treatment right now. So you see, resisting cell death, and we need to so it avoids the cell death, and we need to produce BH3 mimetics. BH3 sensors, you know, they're pro-apoptotic proteins which cause which promote the normal apoptotic signals, and this causes cell death, apoptotic cell death here. Aerobic glycolysis inhibitors. So Warburg effect. So we can interrupt Warburg effect. So when cells have not enough nourishment, um, we have not enough nourishment, and when no more building blocks are produced, the cells are unable for cell division, and that's why we can give inhibitors of aerobic glycolysis, and then cells are unable to produce new new matrix for the cell division. Okay, EGF, so sustained proliferative signaling. So they give us they give a proliferating signaling to this, yeah. And but against it we have EGFR inhibitors. So we also skip this explanation, but okay. But proliferating signals. So we need to block growth factors, growth factor receptors. So growth factor receptors, epithelial growth factor receptors, and then if we have no stimulation of this cells, some of them can die. And immune activating, and avoiding immune destruction, which we discussed in before. So we can reactivate it. We can reactivate it, and actually we already have a treatment against. So you see, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 10 approaches to kill tumor cells. 10 approaches. And usually we use combinations of this combination of these treatments and to kill these cancer cells and to save the patient. But of course, so these drugs, for example, inhibitors of VEGF. So just one example, VEGF is normally present in our body, and VEGF is very important in healing process. Yeah. So for example, in just normal injury, you had an injury, yeah, so to a skin, you need to produce VEGF, and if we administer VEGF to the patient, then it will decrease the ability for healing. So you see, it's dangerous. So then proptotic BH3 mimetics. So also very dangerous chemotherapeutic drugs, because it can activate even stable cells. Even stable cells, they can be killed by apoptosis. So it can be diseased by apoptosis. Okay. For example, I don't know, selective anti-inflammatory drugs, so it depresses the immunity response. So telomerase inhibitors, they can cause the senescence of most of our cells, because some cells in our body normally possess telomerase. And for example, stem cells in digestive tract, or might be in respiratory tract, and they are necessary. We require telomerase in some parts of body, especially in blood forming organs. Yeah. So we need to so for example, blood forming RBC producing cells, they have telomerase, active telomerase, because we need to produce more and more RBCs every day. Yeah. And we don't need to dump the normal cell division there. But telomerase inhibitors, they can cause the decrease of um, anemia. It causes anemia. That's why some chemotherapeutic drugs cause anemia. And actually, if we delve deeper to every of this drug, we can find some side effects. That's why cancer treatment is not ideal now, and sometimes patients died because of cancer treatment, but not because of cancer. So that's why be very gentle with your patients. These patients are uh, your patients are very gentle persons, and you need to care about them very accurately. Okay. We have so what causes the cancer. So as everyone knows this, yes. So I hope you know it before me. So but anyway, radiation carcinogenesis. So, for example, ultraviolet rays or sunlight, radiographs, nuclear fission, radionuclides. So they can cause genetic changes, and every genetic changes, every gene changes, it is followed with tumors. And this example which is shown here, it's Chernobyl. Might be heard about Chernobyl in Soviet Union. It's a very huge catastrophe of the worldwide catastrophe. And when in Soviet Union, so high in this area in Ukraine, in part of Soviet Union before, cancer incidence is very increased. So just because of radiation. Okay, then viral and microbial oncogens. So for example, RNA viruses. So human T cell leukemia virus we have, so which cause adult T cell leukemia or lymphoma. So this it's typical, and actually it's associated with AIDS. Yeah, it is AIDS because AIDS so depress the CD4 positive T cells, and it's so HTLV. So they have a tropism to CD4 positive T cells, and that's causes leukemia and lymphoma. Okay, so oncogenic DNA viruses, we have DNA viruses like Epstein-Barr, human papilloma virus, and Kaposi sarcoma herpes virus. Kaposi sarcoma herpes virus is it's a herpes virus which is associated with, so might be associated with HIV, and they cause the Kaposi sarcoma Kaposi. Now this is mhm. And then polyoma virus called Merkel cell virus. So Merkel cells. So what are Merkel cells? You remember Merkel cells? They are nerve cells of the skin. Okay, doesn't matter. So it's a particular examples. There are particular examples of this viruses which can cause a tumor. For example, human papilloma virus is proved that it causes some of this some types like first, second, and fourth. They cause benign squamous papillomas in humans. They have a very some low risk, especially for 6 and 11, in the genital system. It causes so it might cause a tumor in um, sorry cervix of uterus. It causes cervix uterus cancer sometimes. It's increase the risk. Okay, if I'm not mistaken, that's it. So we have hepatitis B and hepatitis C viruses. They also promote the growth of liver cancer. So usually liver cancer is hepatic cancer is a very rare condition without viruses, but only possession of especially C virus, hepatitis C virus. So it causes formation of tumors there. Helicobacter pylori is a bacteria which causes chronic inflammation in stomach, and this it causes a tumor, so stomach tumor. Okay, clinical aspects of neoplasia. So cancer cachexia. So because of cells, I said before that cells are died, and it causes releasing of a lot of factors like TNF, very commonly TNF, and we discussed it a little bit. Yes, tumor necrosis factors are released, and this TNF causes decrease of metabolic activity inside the body, and it decreases appetite of the patient, and basal metabolic rate is increased, and it sometimes promotes the fever, and that's why it causes cachexia. And then okay, that's one effect. And paraneoplastic syndromes. So these paraneoplastic syndromes are as I said before. So it's when our immunity starts to fight against our tumor cells, but this the same immunity fights against our own cells. So symptoms complex that occur in patients with cancer and that cannot be readily explained by local or distant spread of the tumor or by the elaboration of hormones indigenous to the tissue of origin of the tumor are referred to as paraneoplastic syndromes and also autoimmune. Okay, the most common paraneoplastic syndromes are hypercalcemia, Cushing syndrome, and non-bacterial thrombotic endocarditis. So why it happens? So in some tumors, so it might occasionally it might activize some genes which cause a secretion of um, secretion of glucocorticoids. So then we have okay, so these examples we'll discuss later. And then grading and staging of cancer. It depends on every location. Every location. So this stage grade. We have so that's what is the classification. We'll discuss it in practical class, but it's please read about it. So and then methods of how to detect the tumor. So also we'll discuss it in practical class. So and one of the most useful diagnosis is given by molecular diagnosis. So we can so it's a final so it's one of the last types of diagnosis we have. So actually, it's not present in Kyrgyzstan. It's present only in very evolved countries. I'm pretty sure it's not present in Pakistan too. But soon, I guess when it's a site of research and how to detect these tumors and how to detect, for example, damaged TP53 gene. So how to FISH method and PCR method by PCR method to find these genes which oncogenes, and how to, and it's very it's not important for surgery, yeah, but it's very important for chemotherapy if we want to provide chemotherapy to these patients. So um, it will be necessary to treat these patients. Okay, and this is known as molecular profiling of tumors, the future of cancer diagnostics. So this is if you want, so who wants to get some extra marks and want to be free from exam, so you can prepare SAW. Just talk to your group mates. Only one student can prepare this molecular profiling of tumors, the future of cancer diagnostics, who will be able to be free from exam. So only ones who have good marks in module and in practical class. Okay, on this we stop the topic of tumors. I hope you satisfied with the level of explanation. But if you're not satisfied, I'd understand. I'd agree with you. So if you don't satisfied with the level of explanation, so please read the book in details. In my a lot of details was shut down. Yeah, so I spent 4 lectures, and even in these 4 lectures we couldn't explain everything. So that's why open the book and read about this in more details. Okay, in practical class, I'll ask you about things which we discussed today, and also some points I have mentioned you before. Yeah, so staging of cancer it's also part of discussion. Okay, thank you. So see you next week. So in the lecture class will be next week. So see you that time. Goodbye.
Goodbye,
Let's check
sir.
the attendance here. Sorry, let's check the attendance. Okay, thank you. You can leave.